Good day, friends, and welcome back to the channel. Today, the question is, what are the fundamental tools in a microbiology laboratory? What are the important tools that are used by scientists in a microbiology laboratory? That is, microbiologists. What are the tools they use to carry out experimental procedures and research? These tools, are they easy to understand? Are they the same as what are being used in biochemistry labs and chemistry labs? These are questions to be answered. So what are these tools? Now, number one among these tools are the microscopes. What are microscopes? These are equipment that help to produce large images of microorganisms. Organisms that cannot be seen by the naked eyes are produced. Large images of those organisms are produced by the microscopes. There are different kinds of microscopes that are used in the microbiology laboratory. You have the compound light microscope, the transmission electron microscope, scanning electron microscope. These are all different kinds of microscopes, and the important thing that they do is that they produce images, large images of microscopic organisms, so that these images can help scientists to better study these organisms and study the structures and the functions of different structures within these organisms. These are microscopes. Now, there are also stains and dyes. There are different stains and dyes that exist and that are being used in the microbiology laboratory. And these stains and dyes, what are they used for? The stains and dyes are used to color different microorganisms. Now, these stains and dyes give microorganisms different colors. And the fact being that different structures within the microbial cells absorb these stains differently, thereby creating a contrast within these cells. And also, when these cells are put up on microscopes, this contrast that is being created due to the different ability to absorb these stains by different components of the cells help to create beautiful pictures that can be seen and better understood. There are different kinds of stains and dyes that are used in microbiology. You have the gram stain, which is the most popular stain that is used in microbiology. You have the acid fast stain, you have the capsule stains, you have the endospore stains, and many other different kinds of stains that are used to stain microorganisms. So microscopes, stains and dyes. We also have growth media. What is growth media? Growth media is a preparation which can be semi-solid. If it's semi-solid, we refer to them as agar, or it could be liquid, and we refer to the liquid as broth. So any preparation that supports the growth of microorganisms, especially bacteria, meaning that bacteria are grown on this media, we refer to them as growth media. So if they are semi-solid, we call them agar, and if they are liquid, we call them broth. All these organisms, all these media are used to encourage the growth of microorganisms, and this is quite important to know. There are many different types of culture media. You have the nutrient agar, which is the most popular. You have nutrient broth. You have blood agar. You have chocolate agar. These are all different kinds of culture media that support the growth of different kinds of microorganisms. Also, another tool that is used in microbiology lab are the petri dishes. Now, petri dishes are little dishes. They are little lid-like dishes that have lids, and they could be made of either plastic or glass. And within these little dishes, you have culture media are usually put into these dishes, and then these dishes can be incubated in the incubator in order to encourage the growth of microorganisms. Petri dishes are very, very important. We have some of them being usually 10 to 11 centimeters wide and 1 to 1.5 centimeters high. These little dishes like that, into which culture media is put in, and the microorganisms are grown in these dishes. They are particularly of importance in the microbiology lab. Also, in the microbiology lab, we have test tubes. There are a lot of test tubes, and test tubes are popular. They are well known in science. They are used in chemistry, in biochemistry. They are also used in microbiology. They are used to hold liquid media. Broth are usually held inside test tubes, and they are also used to perform a lot of biochemical tests. There are a lot of biochemical tests that are performed in microbiology lab in order to determine the identity of microorganisms. So many of these test tubes are used to run all these different kinds of tests. So test tubes are important. Also, you have Bunsen burners. These are metal structures to which flames can be put in, and they provide flames that are used in the microbiology laboratory. And these flames are usually used to sterilize equipment. They are used to sterilize the mouths of test tube. They are used to sterilize inoculating loops, and they are used to sterilize different equipment that are used in experiment and experimental procedures in the microbiology lab. Lastly, we have what we call the inoculating loop. Some people call it the inoculating wire loop. And this loop is a small metal, thin metal structure that is used for streaking. That is when the scientist wants to take microorganisms or take samples and streak them onto agar. Streaking means taking the microorganism from the sample or taking a particular sample and putting it, spreading it on the culture plate on the petri dish. So this is what we refer to as streaking, and the inoculating loop or the wire loop is used to perform this streaking. This is just some of the tools that are used in the microbiology laboratory. The microscopes, stained and dyes, growth media, which can be solid or liquid media, the petri dishes, the test tubes, the Bunsen burner, and last, the inoculating loop. Thank you for listening to this lecture, and please remember to like and share our content. Have a wonderful day.